All right, so now I'm going to talk a little bit about yeast. So like I've said already, there are several things that we can do with yeast. One of them brewing beer, and one of them is baking bread. So yeast is, importantly, what we call a facultative anaerobe. What's that mean? Well, it means that, remember, aerobic organisms and anaerobic organisms, or rather aerobic respiration and anaerobic respiration. So yeah, aerobic respiration is when we have oxygen. So we talked about that. That was topic 12, I think. And anaerobic respiration is respiration without oxygen. So it turns out that if you're a facultative anaerobe, you can do both. And actually, we are facultative anaerobes as well, or rather, our muscle cells are. So if you remember what our muscle cells do, they use the oxygen when they can, and if they don't have enough oxygen, if oxygen isn't supplied at a high enough rate, they will switch to anaerobic respiration and make lactic acid. Yeah, so hopefully that will ring some distant bells. So it turns out that yeast is just like our muscle cells, except that it's a different type of respiration. Yeah, so when there is oxygen, then yeast is going to perform aerobic respiration, so burning glucose all the way down to carbon dioxide and water. If there is no oxygen, yeast has the possibility to switch to a different type of respiration, so that would be anaerobic respiration, and in particular alcohol fermentation. So yeast will then start with a molecule of glucose and burn that down to ethanol and carbon dioxide. Yeah, so you get two products, ethanol and carbon dioxide. That is important to remember. You give it glucose, if it doesn't have any oxygen, it will produce ethanol and carbon dioxide. So you've got both the word equation at top and the balance symbol equation at the bottom for that reaction. Now, why is that important? Well, you can use yeast to produce ethanol. So the brewer's yeast or a baker's yeast, Saccharomyces cerevisiae, that's its name. It is used in brewing. And where in brewing you give it sugars from malt. Now, what is malt? You probably all know that there's malt in beer, but do you know what it is? It is when you take cereal grains, you have them germinate and then you dry them. That is what we call malt. So there's malt from different types of cereal. Yeah, malt is not a cereal. Malt is after, well, it's basically what you get after you've done a certain process to any cereal which involves germination and then drying. Anyway, so this malt, you have to make it germinate because otherwise there won't be any sugars, otherwise there will just be starch in it. But once you've got the sugars after having the cereal germinate, then you basically feed those sugars to a yeast, and the yeast, you have to do that under anaerobic conditions. And as a result, the yeast produces ethanol, and that's where the alcohol in your beer comes from. Well, not in your beer, obviously, but that's where the alcohol in beer comes from. When the alcohol content is too high, it becomes toxic for the yeast. And that's, I think, as I said earlier in the previous part of this presentation, that's where you've got a limit to the alcohol content of beer. So that's one use, beer brewing. Another use of the same metabolic pathway is when you're using yeast in the production of biofuel, which is nothing else than ethanol as well, from different crops. So you can use corn or sugarcane, and it's the same type of fermentation. You're starting off with glucose and you're ending up with ethanol and carbon dioxide. Well, in this case, the carbon dioxide isn't put to much use other than it makes your beer, again, not your beer, but it makes beer a little bit less flat. Yeah, it makes it a little bit effervescent. All right, so then you can also use yeast in baking. So in bread making, you still get ethanol and carbon dioxide, but instead, in this case, what you want is actually the carbon dioxide. So the alcohol, you get it as well, but when you put, after having the bread rise, which is where the yeast sort of gets to work, you put the bread in the oven and the ethanol evaporates. The CO2 makes the dough rise. Yeah, so you have to do this for an optimum amount of time If you don't let the bread rise enough, it's going to be sort of flat and quite dense. If you let it rise too long, then you get too many holes in your dough, and that's not nice either. Okay, so during baking, what happens, other than lots of chemical reactions between the different components of the dough, is that the yeast die and that the ethanol evaporates. Yeah, so in that case, you will only keep the CO2 from that process. Yep, so remember, for brewing, it's the alcohol that you want. For biofuel production, it's the alcohol that you want. Same process for bread making, but here is the CO2 that you want. Okay, so now we've talked a little bit about biotechnology and the different types of uses that you have for microorganisms, but we haven't modified the microorganisms yet. We haven't actually manipulated them that much, other than growing them in large fermenters. Now we're going to talk a little bit about genetic engineering. And in genetic engineering, per definition, the genetic material of an organism is changed. How can that be done? Well, you can either remove or insert individual genes, or you can also change them in some way or another. Why would you want to do that? Well, there's various reasons. There's actually many reasons. Here, as part of your syllabus, you're going to learn about two different types of, or two different situations where genetic engineering is used. One is the insertion of human genes into bacteria to produce human insulin. So if you think about this, this is quite extraordinary. Yeah? So you take a human gene from our genome and you somehow get it inside the bacterium, and then you rely on the whole cellular machinery of the bacterium to make insulin, which is a protein. It's a short protein. It's a very short protein. We actually call it an oligopeptide rather than a protein, but it's the same thing. It consists of amino acids, so it can be produced, just as it would in our body. It's being produced by a bacterium. So that's crazy, right? But it's possible. So that's one example. The other example is the insertion of genes into crop plants. Why would you want to insert any genes into crop plants? Well, if you're a farmer, then you would want your crop to be resistant against herbicides and insect pests. Yeah, herbicides, why? So that you can spray, this is a whole other discussion, should we do this or not? But if you're like an industrial farmer, you want to just spray your crop, and you want, so to get rid of weeds basically, and you want the crop plant itself to be resistant against the herbicide. And insect pests, well, that's quite obvious as well. There are lots and lots of insects which can really wreak havoc on the harvest. If you could just, without having to spray an insecticide, if you could get rid of these insect pests, that would be fabulous. And that can be done by transferring genes that produce pesticides into the plants themselves. So they produce it rather than you having to spray them with it. Okay, so there's a difference between a herbicide and an insect pest. Oh, sorry, there's a difference between herbicides and insecticides. Herbicides kill weeds and insecticides kill insect pests. Also, you might want to insert a gene into a crop plant so that you can make the plant produce additional vitamins. So that's often done as well. There is the example of a variety of rice that is called golden rice, and it makes vitamin A. And that is then used because rice is actually naturally quite poor in vitamin A, and many parts of the population of countries where rice is, a widely spread staple food, don't actually get enough vitamin A. So one way of dealing with this is to make the rice produce vitamin A, and then people eat it, and people don't have a vitamin A deficiency anymore. How do you do that? Yeah, so here we're going to focus on the insulin example. So we're going to take the human insulin gene and we're going to insert it into a bacterium so that the bacterium will then produce it for us. The method being used is called recombinant DNA technology. So what do you do? So on the scheme on the right hand side, you've got a bacterium with a plasmid. And you also have, more towards the right, you've got a cell DNA containing your genes. In this case, it's the insulin gene that we want. So first of all, you'll start by, that's the first point, by isolating a bacterial plasmid. We call that a vector in this case because it carries something from A to B. Yeah, it will carry the gene into the bacterial cell later on. So that's why we call this kind of plasmid that we use to carry genes a vector. You will then take a special enzyme, which is called a restriction enzyme, and you have to imagine it like a pair of molecular scissors. These scissors manage to cut open the plasmid. So literally, it's a molecule, you've got plasmid molecule, it's a circular molecule of DNA, and you cut it open with these molecular scissors, which are an enzyme. Then you take DNA from a human cell and isolated the gene that you want, so in this case the insulin gene, from the human cell. Again, you will cut it out of the genome using a restriction enzyme. Okay, so here again you're using an enzyme to cut down a DNA molecule. Then you mix the two. Yeah, so I'm now at stages, we've done stages 3 and 4, and we're looking at stage 5 on the figure. So here I'm going to mix the plasmid that I took from a bacterium and the human gene. This is really, really tiny, of course. Yeah, so these are really molecules, very small, but we can do this. And you mix the two, you've got them in solution, and you just use a pipette to mix that solution. That's how it works in the lab. And they will, you know, the human gene will sort of nestle itself into the plasmid. Now, how do you make it stick there? Well, for that you need another enzyme. So the enzyme ligase, which sticks the human gene into the bacterial plasmid. So that sort of mends the gaps that, you know, if you look at the plasmid in picture 5, you'll see that there are small gaps between the plasmid and the human DNA. And then you throw ligase onto that, which is a, you know, sticking enzyme. And those gaps in picture 6, they're gone. That's because now the plasmid and the gene of interest, our insulin gene, are ligated. These plasmids, now carrying the insulin gene, are then taken up by bacteria, and you just, you know, you grow your bacteria in a big biofermenter, as you did before. And then the bacteria will produce human insulin for you by just reading off the plasmid and making the insulin just as our human cells do that. Okay, so what advantages to that? Seems fairly complicated, but it's actually really great because your product, if you have it produced by microorganisms, is going to be really pure. Yeah, rather than, so what people did before having microorganisms, they used pig insulin, and you can isolate that, but it's going to be less pure, and also, that's the second point, it's going to be pig insulin, which is, you know, it works, but it's not as good as human insulin in humans. So having our human product produced by microorganisms gives it an edge over the pig insulin. So that's another advantage. Then also, using this method, you can produce quite large quantities, and you can also easily adjust your production to demand. Yeah, so whenever you need to produce more of insulin or anything else that you're producing that way, you can just fire up your biofermenter and you know, make the bacteria reproduce and produce your product. And in times where maybe the demand isn't as great, you just you know, freeze a little sample of those bacteria, put them in your freezer, and they will live there happily. And you know, whenever you need them, they will be available. Okay, one last thing here is gene therapy. So the method that I just discussed, so this insertion of a gene into what we call a vector, so a construct that will carry a gene from A to B, that's what we call a vector in biology. That principle can also be used, or at least in theory, be used to transfer genes into human cells, not just bacterial cells, but into human cells. So why would we want to do that? Well, imagine a person which has a mutation in an important gene. And maybe because of that mutation, the important gene doesn't do what it's meant to do anymore. And that can give rise to genetic disease. An example of that is cystic fibrosis, yeah, where people have a problem with a particular gene called the CFTR gene. And this is a recessive condition, so you need to be carrying 2 mutant copies in order to be affected. But if you do, so if you do carry the 2 copies, and if you are affected, then you're going to be producing a very, very thick mucus that clogs up your lungs. And there's other problems as well, but that's one of the main problems. Now there are, you know, sort of manual therapies for that. You can sort of do this therapy where you tap your chest and that will loosen up the mucus and then you can cough it up, but it takes a long time, and anyway you are at very great risk of infection and so on. So that's really it's quite a dangerous condition, and people with cystic fibrosis they're often in hospital fighting infections and so on. So wouldn't it be great if we could introduce a normal copy of the same gene into those cells? We're talking about lung tissue here actually, and then have this normal gene make a normal protein which could restore the function of those cells. Well, that's exactly what people are trying, and there are various approaches. So in all of these approaches, what you're doing is you're taking the normal gene. Now you've cut that out of somebody's genome. You extract DNA, for instance, from blood cells or saliva cells or something like that. Whatever is a good source. And then you take the human gene, the sort of normal one, the one that doesn't have a mutation from a donor, and you stick that into a virus. So here our vector isn't a bacterial plasmid, because that wouldn't go into a human cell, but viruses will. Okay, so viruses are obviously in the news now, and we know that viruses go inside cells. So take a virus that is, happy to enter a lung cell, stick the gene in there, and make the virus go into these lung cells, and it will deliver the normal gene to the cells that need it. There are various approaches here. One is called integrating gene therapy, where the gene is permanently inserted into the patient's genome. One is called non-integrating gene therapy, where the gene is still inside the patient's cells, but it just does its own thing without being integrated into the patient's genome. And finally, there's something called RNA therapy, where you give messenger RNA, remember that, instead of DNA. All of these are under clinical, in clinical trials at the moment. Some have given promising results, and none of them are really fully established yet. But it's definitely an interesting approach. Yeah, so that brings us to the end of this topic, and here's just an overview of the different concepts that you should have learned about, that you have to know about. You'll find them in the sections of the book that I've given you at the beginning of these presentations. So here they are again as a reminder. Yeah, so you've got section 20.1, Biotechnology and Genetic Engineering, sort of an overview. Then you've got Biotech and Genetic Engineering. All right, and that is the end of the topic.